thank you, Vincent, for the nice introduction, and thank you all for staying around here on a Sunday afternoon. And so my name is Julian de Ruiter, and I'm currently working as a data scientist at GoData Driven. But at my previous job, I worked as a PhD student at the Netherlands Cancer Institute, or the NKI, and essentially my research focused on researching breast cancer and with the genomics data. And so one thing I noticed when I was talking with my new colleagues is that if you talk about machine learning in the medical field, that people very quickly jump to images, which they can easily relate to. But for genomics data, this is much more abstract because people aren't used to thinking about these kind of things. And that's why I thought it would be nice to, maybe at a meeting like this, to introduce some of the concepts here and give you an idea of what it's all about. So that's why we're here today. And I'm giving this talk together with Nanne, who's a former colleague of mine, also from the Netherlands Cancer Institute. And this talk will essentially consist of two parts, where I'll first try to give some biological background and some introduction into the world of tumor sequencing and what kind of data you get from this. And then Nanne will take over in the second part on explaining how you take data from these sequencing approaches and use them to create machine learning models. For example, in this case, focusing on predicting drug responses, which is one of the main topics in his thesis. So first I wanted to start very broad, so with some biology. And as many of you might already know from some biology course in your secondary school, our body essentially consists of, I think, around 40 trillion of these very small things called cells. And each of these cells is governed by a long strand inside the nucleus of these cells called DNA, which if you zoom in on this, it essentially is a long double helix strand which contains many functional elements called genes. And what these genes do, they essentially encode for the building blocks of the cell called proteins. And what happens inside the cell is that first these things are read off by some cellular machinery in a process called transcription to create a messenger molecule called RNA. And this is then later moved to somewhere else in the cell and then translated in a process called translation into proteins. And proteins are essentially one of the most important biological building blocks of the cell because these actually do things in the cell. And this is a quite tightly regulated process which always ensures that for a given gene you always have the right amount of protein that you have in the cell for performing a particular function. And what's important to realize is that these proteins don't act in unison, but they act in quite complicated networks. And I just wanted to give one example of a signaling network, which is often used, often shown. And in this case, you can, for example, imagine, that there's a protein which acts as a receptor on the outside of the cell, listening for some kind of signal. And when it encounters a signal, in this case indicated by this block EGF here, it relays the signal inside the cell to another protein, and then again to another. And this kind of signaling cascade at some point then relays to the inside of the cell, some kind of signal indicating that it should keep on surviving, keep on growing, etc. And while this is quite easy to understand because it's just a linear cascade, you also have other interactions where you, for example, have other proteins which then act as a brake on this on the proteins in this pathway, so blocking the growth signal. And you also have more complicated things where you have signaling pathways in parallel in the cell, which then also sometimes interact with each other, and basically resulting in quite complex networks which show quite complex behavior. And this is just a very simple example, because if you plot all a network of all these proteins and their interactions, it quickly looks something more like this, where you can see that there's a lot going on. It's much more difficult to understand what's actually happening in a network like this. So then very briefly, what is cancer? So if everything's going all right in our body, cells work together to create these complex structures, in this case like kind of a skin structure, where they form nice rows and then basically do what they should do to create a proper tissue structure. And what sometimes happens during the course of our life, or during the course of a life of a cell, is that they accumulate mutations, either because we've been sitting out in the sun too long, or been smoking, or stuff like that. And these mutations essentially then, if we then project that back onto a network like this, we can imagine that a mutation in this case might, for example, knock out these proteins, so make it non-functional. And as a result, this signaling pathway that I showed is no longer controlled or repressed by this protein, which make this much more active than it should be. In this case, signaling that the cell should just keep on growing, keep on growing, keep on growing. And what then happens in this case, or can happen, is that this cell with the mutation can keep on dividing and create this kind of mass that then displaces normal cells and ruins the tissue structure locally. And if this continues further into the tumorigenic process, you get this behavior where the tumor cells start invading into the surrounding tissues. And if you're unlucky, they also start segregating off and migrating to other parts of the body, which is a process known as metastasis. And of course, if we look at the clinic, one of the important questions in cancer research is if a patient comes into the clinic and they already have cancer, what is the best approach forward? So what's the best way to treat this patient to ensure that they recover from this? And so traditionally, one of the more used approaches for this is to give the patients chemotherapy, or in the case of breast cancer or prostate cancer, you also have some hormonal therapies. And if you're lucky, these therapies can have a very good effect and eradicate the tumor, which is the best case. What you see very often is that tumors also sometimes they do regress or give some kind of stable disease. So this essentially prolongs the life of a patient but doesn't really cure them. And if you're very unlucky, what happens a lot of the time is that tumors just keep on growing and the patient dies eventually. And one of the surprising things about this is if you look at this, patients often come in with quite similar tumors, but some patients respond very well to chemotherapy or to some kind of therapy, whilst other patients don't respond at all. And therefore, one of the really key questions here is, can we identify why some patients respond to treatment whilst others don't? And can we use this information to maybe devise new treatments or assign patients to the right treatment groups? And this essentially one of the main ideas behind an approach called personalized medicine, where the idea was to, when a patient comes into the clinic, to take a biopsy from the tumor of the patient and then feed this into a big machine called a sequencing machine. And what this machine essentially does, it allows us to measure some features on the genome or the genetic side of these tumors, such as mutations, how strongly genes are expressed, and how often they are contained in the genome. And then the idea behind this is that we can use these features to predict if they will respond, for example, to specific drugs based on the mutations they have, maybe also identify some tumor-specific vulnerabilities which we can use to devise new treatments. And so just to give a brief introduction to how tumor sequencing works, the idea is that we use some kind of lab process to extract the DNA from the cell and feed this into the sequencing machine. What the sequencing machine essentially does, it generates many, many short sequence reads, which are about 50 to 300 base pairs long. So each base pair in this case indicated by an A, C, a G, or a T. And so like I said, each sequence is around 300 long. And then to make sense of where these reads came from, so from which genes of the genome, we typically align these back to a known reference genome, which was generated, I think now 10 years ago. And just to give some idea of the scale, this genome is around 3.2 billion base pairs long. So these are really short sequencing reads. And once we do this with a text alignment algorithm generally, it looks something like this, where we take these sequencing reads and map them onto the genome. And then once we have these mapped reads, we can then look at the positions at each genetic position along the genome, and then compare the sequences that we see in this tumor or in this tumor sample to the reference that we know. And in this case, you would see that the letters in each position is the same, except for here we see a C instead of a G. And this might indicate that this is a mutation in this tumor, which might have an important biological impact on the cell. At another level, we can also use the RNA, which I mentioned briefly earlier, and stick that in the sequencer, which also gives us a lot of sequence reads, and we can also align these to the reference genome. And in this case, because this is basically how high cells expressed, we can then align these to the genes in the genome and then count how many sequence reads we have at a specific gene in the cell. And this gives us some kind of idea of how strongly a gene is activated in these cells. So in this case, because we have a lot of sequence reads mapping to this first gene here, it would have a high expression level, whilst because we only have a few reads here, we would have low expression for gene B. So this is a bit basically the basics behind tumor sequencing. And some challenges in analyzing this kind of data is that it's quite a complicated multi-step process where we do a lot of different steps like contaminant filtering, the alignment I mentioned, but also a lot of quality control and also downstream things like the mutation calling. And beside that, there are a lot of different tools involved. They're often written in different languages because it's all academic software, and they have a lot of complex dependencies, which makes them difficult to install as well. And what's also very fun is that you have a lot of tools doing the same things, but they often give very different results, which has led to some cases, for example, where people have advocated for mutation calling to just run 5 or 6 different algorithms and take some kind of weird intersection of the results. And this makes it difficult to kind of um, formulate this process cleanly. And also, maybe on the logistical side of things, these processes also produce many large files, which are typically 10 to 100 gigabytes per sample, which in a hospital environment can be quite challenging to store efficiently. So given this complexity, at least in my job, I thought it was very important to try and facilitate the reproducible analysis of these kind of genomics approaches. And to do so for my own work, I use two basic tools. One is Bioconda, which is, for those of you who know, Anaconda is essentially a repository, an Anaconda repository, but then for bioinformatics tools. And they also have a lot of bioinformatics-related Python R packages, which makes it very easy to properly define your software environments and make them reproducible elsewhere. And beside that, I also use a tool called Snakemake, which I'll go into in the following slides, which I essentially use as a workflow management tool for defining the different tasks and creating analysis DAGs. So just to give you an idea what the Snakemake looks like, it's essentially a DSL, which generates Python code in the end. And this is just what a simple rule looks like, where a rule has a name, in this case a line. It takes an input, which is a certain file. It generates an output, which is also a file name in this case. You can add certain extra parameters, which don't have to be files, but in this case, this is also a path. And it can also then use these inputs outputs to generate, in this case, a shell command, where I call a tool called BWA with this index, with this input, and then generate a certain output. And of course, it's useful if you're just generating one file, but Snakemake also allows you to put wildcards into your input and output, as many as you want, which allows you to generalize this rule. In this case, if I want to then generate a SAM file with a certain sample, it knows that it has to interpolate this name into the input to find out which input file it needs. And besides running certain shell commands, you can also, it also has good integration for Python or for R scripts, and you can also run arbitrary Python code in here if you want to. And some advanced features that I also like, which I just wanted to briefly mention, is that you can also define remote files. In this case, I show an example for S3, where it can, as an input file, you can specify a file for S3 to use, and it will automatically check if this is updated and download it if needed. You can tell Snakemake that some files are temporary, in which case it will just throw them away as soon as they're no longer needed, which is nice for these big file sizes. You can define the resource that a certain rule needs in terms of threads, but also memory or anything you really want. And you can also, per rule, you can define a certain environment to use if you have a specific software version that you need to use. And this can be a Conda environment, but there's also support for Docker Singularity if you want to go through the container route. And what essentially it looks like if you put this all in a big Snakemake file, you start with the kind of default rule, which tells Snakemake which files you want to generate. In this case, I'm asking for two SAM files. Then it looks for rules to generate these files if they're not present in the file system. In this case, it finds that it's a line rule can generate these files, and it needs this input. And then it looks further to see if it can generate this input with a different rule. And then it finds this rule, which has this as an output. And then in this case, this would be my input sample, which is already present. So this would kind of define the entire DAG. And what it looks like when you want to execute something is that these rules are essentially used to build a DAG of tasks. And this is just a dependency graph for a simple DAG which only does the alignments and some QC. And arrows in this case indicate a dependency from this job on the previous job. And tasks are executed as soon as the preceding task is completed. So in this case, this merge task would be executed as soon as this one succeeds. And resources in that case are taken into account. For example, if a job needs a lot of threads and they aren't available, it will wait to executing that job. And what's also very useful is that it can run these jobs in parallel locally. It can also use a cluster if you have a cluster environment, or it can also run in Kubernetes cloud environments, for example, which I haven't tried, but I think is very useful. And the previous graph was just showing some dependencies, but this is essentially then for what the entire DAG would look like for so 4 samples sequenced over 2 sequencing lanes. So you see that it explodes very quickly in terms of the number of jobs that you have. And the analysis I typically use this for is more in the order of hundreds of samples. So it gets big very quickly, which also indicates why I need software to orchestrate these kind of things. And the output then of this DAG that I showed very briefly is that it would generate alignments, and that these you can then use in further processes to generate like these mutations and expression counts, which can then be used in downstream models, which Nanne will talk about in the remainder of this talk.
All right, thank you very much. So what this gives us is basically two feature matrices with samples on the rows and features on the columns, one for DNA and one for RNA. And as Julian has alluded to, we want to relate those to treatment response or drug response. So on the same set of tumors, we also have some form of drug response. Typically, we have a continuous measure, namely the concentration at which the tumor starts to respond. Now, to relate this to each other, we can use, we can try to predict the drug response to anti-cancer drugs, and we will do this by phrasing that as a regression problem, where X will be defined as the concatenation of those DNA and RNA data into one big feature matrix. Our drug responses are Y vector, and we'll relate that to each other via linear regression, so fitting a vector of betas to map X to Y. We've also tried other nonlinear methods, but those don't really add any predictive performance over the linear case, so we're going to stick to the simplest model here. As you probably know, we obtain our betas by optimizing this function. From here, I want to focus on a couple of very specific disadvantages or challenges, really, that we have in this kind of data. Number one, for example, is that we typically have way more features than we have samples in these cases. So for the data that I've shown here, or that I'm going to show here, we have way over 20,000 features versus only about 1,000 samples. So we're working with very high dimensional data. And one way to deal with that is by using regularization. So for example, using ridge regression, which means that we add a term to our optimization problems to sort of prevent these betas becoming very, very large, which typically doesn't generalize very well to other settings using ridge regression. Another challenge that's specific to this data is that we would like to make interpretable models, and a linear model with 20,000 regression coefficients is not very interpretable because we still don't really know which genes are related to or responsible for the drug response. And while we actually want to get biological insights from this data, because also when we sort of biologically the model makes sense to us, we might also trust its predictive performance more. The way we're going to do that is, or that we did that, is by using feature selection. And as it turns out, when you have these sort of penalized regression models, there's a penalty that sort of at the same time performs feature selection as you optimize the regression model. That's done by adding this L1 penalty, as it's called, so summing over the regression coefficients, or the absolute value, actually. This is when you combine these penalties, they're called elastic net regression. I'm not going to explain exactly why this works like it works, but I'll give an example of what it looks like. If we have our feature matrix again, X and Y, and we have a beta coefficient, then most of our regression coefficients, when we optimize this formula, will be 0, so they won't be selected. They will be sort of like shrunk to 0. But a small number of regression coefficients will be non-zero. They will actually be used in the prediction, and those are then your selected features. So up until a couple of years ago, this was what many people used to predict the drug response from DNA and RNA data. And when we applied these models to this particular kind of data, we found that there was another sort of challenge, which is that there's strong correlations between these data types. DNA and RNA are very correlated to each other, and simply concatenating all of that data into one big matrix X and performing your machine learning might not be very optimal. I'll give you an example of that. Before I do that, we'll use a slightly different data set with a few more data types, namely this data set where we have mutation, CNA, cancer type, methylation, and gene expression data types. What those are exactly is not super important for the rest of the story, but if you're interested, the first two are sort of related to your DNA measurements, and the last one is related to your RNA measurements. Now, if we train this complete model and we get a regression coefficients beta, and we're going to do this for a lot of different drugs, so we have about 265 drugs, and hence also 265 models, we can visualize those models as follows in this plot. We have our same 5 data types on the x-axis and something I call the relative contribution of each data type on the y-axis. That's based on our betas, our regression coefficients. Each point in this box plot corresponds to one of those models, one of those 265 drugs. And what we can see here is that almost for more than half of the models, or most of the models are almost completely based on gene expression. The median contribution of gene expression is 100%. And this is a bit weird. It could mean that maybe these other data types are not predictive of drug response at all, but that's actually not what we see. Because when we plot these 5 data types again versus the predicted performance of an individual data type, we see that while gene expression is the most predictive, these other data types are actually also kind of predictive. So this got us thinking, like, what could be going on? And before I'm going to hypothesize about that, I see actually I have one more slide here, which is when we think back about that combined model that we had, and we looked at the predictive performance of that model, surprisingly enough, we see that it actually has virtually the same predictive performance as our gene expression-based model. So the reason that we think this is going on is as follows. Remember that these data types are correlated to each other, they're related to each other. And what we sort of know from what also our prior biological knowledge is that some of these data types, mutation CNase, cancer type methylation, I'll call them the upstream data types from here on, influence the gene expression, through which they may influence the drug response. So in other words, we think that the upstream features affect the drug response through the gene expression. And that of course means that the gene expression is more correlated to the drug response, which then it makes sense that a feature selection algorithm would also select most of these gene expression features. But it's also true that we're very much interested in these upstream features because those are often causally related to the drug response. And they're more interpretable in that sense if we select those. So can we somehow sort of like prioritize the selection of these upstream features? The way that we're going to do that is very simple. Remember that our previous model combined all these data types. I put them into one big feature matrix and then predicting the drug response. What we're going to do is a 2-stage approach that we've called tandem, where we first take the upstream data types, explain as much as possible of the drug response using those data, and then in the second stage, we're going to explain the remainder using gene expression. So we're predicting the residuals in the second stage. And to show you that it works, on the left, we have the plot that we've seen a few slides ago, where we show that this classic approach that people have been using selects almost exclusively gene expression features. Whereas in our approach, we select, this is reduced by about 50%, with the remaining 50% attributed to the other data types. So we have way more selection of upstream features. Of course, we need to check that this is not at the expense of predictive performance, and indeed it isn't, because we plot the predictive performance using the classic approach versus the predictive performance using tandem. Again, each point is a drug here. We see that this is highly correlated, and there's virtually no identifiable shift here. So just to show you what this increased interpretability means, I give you an example. So Julian has shown you some of these networks, and this is one how these genes supposedly work together. This is one of the drug targets. This is sort of like inhibited by the drug. All the green stuff is selected, or the light green stuff is selected by both the classic approach and by tandem. But our approach, tandem, also selected 2 additional ones, the dark green ones. And when we started to think about those biologically make a lot of sense to us. With that, I hope that I've shown you a nice overview of how we used our bioinformatics and machine learning to go from cells to drug responses. Before I end, I would like to show you some of what we think are future directions in this field. Sample sizes are ever increasing, and this is mostly because the cost of sequencing really decreases rapidly. If you look at this graph here, you can see the price on the y-axis of sequencing a human genome versus time. And note that this is actually a log scale. The price is on a log scale. So some people have suggested that the price goes down faster than Moore's law, which sort of suggests that at some point we're not even going to be able to keep up computationally wise with the amount of data that we're producing here from sequencing. This has also led to lots of public data sets, ICGC, TCGA. Together those contain more than 10,000 tumors right now, and more are probably to come in these kind of projects. Another cool sort of development is the introduction of nanopore sequencing. Remember this big machine that Julian has shown on his previous slide. Now they are working on very small machines that you can plug into your laptop, and you can actually start sequencing small sequences at home. These things start at about $1,000, so those are pretty cool. So we might be seeing more and more of those sequence data around. In terms of challenges, not all is solved. Tumors are very heterogeneous. They start as single sort of clones with one or a couple of mutations, but then they mutate into another clone and a different clone. And your treatment might kill one clone, but not the other. So tumors often become resistant to the treatments that we're giving them. So ideally, we should be sequencing at multiple places in patients or over time to see how do these tumors evolve and what kind of different clones do we have and so how does that affect treatment. And of course, these sort of different samples per patient also affect our statistical models and how we should think about those. And finally, here we've shown you sort of like an approach that takes all of the genes together and simply uses regression to predict the drug response. Another approach would be to sort of incorporate prior knowledge and sort of go bottom up, starting with these networks and slowly adding genes that we think are relevant. And this is often done by computational, often Bayesian modeling of gene networks within tumor cells. Those were our perspectives on the future. Thank you very much. I'd like to wrap up, and we're happy to take questions.
We have about 10 minutes for questions, so, well, 8, but there's plenty of time. Yes.
Yeah, I was wondering a quick question. So you are talking about the treatment response. Is that a binary variable?
So the question is whether the treatment response is a binary variable. In this case, we're looking at response to the drug, where the response is indicated as a continuous variable, namely the concentration at which the tumor started responding. And maybe as a slight addition to what I've told you here is that the data set I've shown you here is actually something about tumors that are kept in the lab. So they're outside of a patient and they grow in a dish in the lab actually. So that's why we can test different concentrations and actually see which concentration did the tumor start to respond. You're absolutely right, and in typical clinical situations, if you've got data from a clinical trial from patients that have been treated, you usually look at either binary response rate or some ordinal response rate of response, partial response, or no response.
Yeah, because I was also wondering there are large parts in the DNA that are never activated. So how do you identify the parts that are likely not interesting and should not be included in your model?
So the question is, there are large parts of the DNA that are not relevant, and how do we identify those? So we're focusing on the parts that are genes. So that's already just about 3% of the entire genome. And of that 3%, those genes, most are in some way typically relevant. Not to every drug, but that's why we're doing the feature selection.
So those are the multiple ways to correlate these things. You can either look at genes that are often mutated in certain cancer types, which is more like just looking at mutation frequencies. And also people often use prior knowledge in this case as well, where they know that a certain gene plays an important role in certain processes. But also, just letting to your remark about that a lot of the genome isn't important, that's not entirely true. So like the remark that DNA consists of only genes is very simplifying. It's a bit of a simplification, because there's also a lot of research going into all kind of sequences that are not coding for genes, but do have a lot of effect on gene expression via all kind of feedback and regulation mechanisms. So this is more or less some of the simpler stuff, and there is a lot of biology underwater that is a lot more difficult to understand. But also, one of the issues here is if you find mutations in a gene but you have no idea what the gene is doing, then it's not very easy to do anything actionable with, because you have no idea what kind of drug you should develop to target this, or what the gene is actually doing biologically to the tumor.
I'm also wondering what the influence of the phenotype is. So age, weight of the patient, gender, things like that. And are there any studies on that?
So the question is whether age or weight or things like that matter to the response in these clinical studies. So not the ones that we are doing with the cells in the lab, but in clinical studies, those are typical parameters that are taken into account. And age is very predictive of whether someone is gonna make it for another 10 years, say. Because if someone is already very old, they might die sooner, or more likely. I work with data from the lab, which is why my phrasing was not very nice just now, I guess. Sorry.
Yeah, but my idea is more like, why are you focusing on genes and not on other
Well, the genes do have the question was, why do you focus on genes and not those other things like weight, etc.? There's definitely lots of research there too, but the reason to focus on the genes is that typically tumors originate by genetic defects. So the rationale is to also target those defects using drugs, and that the information of which drugs will work where is sort of like related to those genetic defects. So that's why we're looking at the genetic data there.
And I think a lot of clinical variables like weight, age, have a strong influence on what kind of tumor you develop. Like breast cancer is typically in older women, for example, after menopause. But not necessarily, like Nanne said, how a tumor responds or how it originates and how it responds to treatment is more defined by the genetic features. And that's why after, well, you could determine for a long time how old someone was and try to correlate that with their treatment response. But like doing this sort of genetic approach is much newer, and this adds to that approach basically. And I think you can explain much more from the genetic side of things than from just looking at clinical variables.
Anyone else have a question? That's up to you.
Stein?
Yes, so I have never heard of the tool Snakemake. How does it relate to things like Airflow and other scheduling software for Python?
So one very big difference with
How does Snakemake relate to?
So the question was, how does Snakemake relate to, for example, other workflow tools like Airflow? And so one big difference, I think, is that Snakemake is more like, well, GNUMake, for those of you who remember GNUMake. And in that sense, it focuses a lot on files, and it focuses a lot on, it starts with what you want, your results, and then traces it back to what you, through the rules that it has to its inputs. And it's very much file-oriented compared to Airflow, where Airflow is much more time-oriented, where you schedule jobs to run at specific time intervals. And this is more like I get a data set with a certain set of samples, and I want to generate a predefined set of outputs from these samples that I get. So in that sense, it's, yeah, very different, I think.
Time for one more.
Maybe this is a very basic question, but I was wondering if you have a lot of features but not a sample, would it make sense to use the predictive component analysis to reduce the number of features and club the correlated features together?
So the question is whether, because of the high dimensionality problem, we shouldn't, or we could try to use principal component analysis to reduce the dimensionality and go from there. People have done that. It works well. It doesn't necessarily work better than the approach we discussed here. The predictive performance is roughly the same. And the major advantage of doing feature selection immediately is the interpretability, because you're really looking at a small set of genes that are related to your drug response rather than a linear combination of, once again, actually all of the genes.
Okay, fine, one more.
Because tumors can become resistant to the treatment.
Mm-hmm.
Then you would have no dose where the tumor reacts to how you handle these cases.
The question was, what do you do when you don't have a dose at which the tumor responds or reacts, and how do you handle that case? Because you have to do some sort of extrapolation. Well, that's actually the answer. We do some sort of extrapolation. We realize that there is some inherent noise to that because, well, you're extrapolating and that's not necessarily easy still. We've tried some other approaches like just binarizing it, but there is a difference between responding a little and responding a lot, so we do sort of prefer to use the concentration in the end. And still, these extrapolations, they all tend to extrapolate their response in the resistant part at least. There are some things that you can do that we didn't really discuss that are probably well suited for offline discussion because they go into curve fitting really. All right.
With that in mind, they will still be around for this small intermezzo, but let's give them one last round.
Okay.